The steroid study everyone's been talking about, but they might be getting it wrong. Hey everybody, welcome back to my channel, and today I wanted to walk you through a paper that genuinely caught my attention when I first read the title. It looks at anabolic steroid use, training style, and molecular markers related to muscle growth in competitive strength athletes, a combination we almost never get to see studied directly in humans. Now, research in this area is extremely difficult to conduct, largely because anabolic androgenic steroids are illegal in most countries outside of medical use.
And remember, hugs, not drugs.
That makes controlled human studies challenging to conduct, and as a result, much of the existing evidence comes from animal models or short-term lab-based interventions that don't always reflect real-world training. So when I saw a study that included competitive powerlifters, bodybuilders, and strongman athletes, I was genuinely intrigued. Now, as I worked my way through the paper a little more carefully, that excitement became a bit more tempered, not because the question isn't important, but because the methods used in this study impose some limitations on what the data can actually tell us. Now, this paper has been widely shared on social media, yet many of the interpretations I've seen have missed some important context. So let's take a closer look at what this study actually tells us. The authors examined the expression of 3 genes that are often discussed in relation to muscle growth and remodeling, specifically IGF-1, MyoD, and myogenin. However, these were measured in the present study using salivary mRNA, not muscle tissue. That distinction is critical for how we interpret the findings of this paper. So rather than examining what's occurring directly within the muscle fibers, this study investigates whether long-term training style and steroid use are associated with differences in peripheral gene expression, so that is, gene activity in tissues such as muscle, fat, and blood cells measured via saliva. All that to say, this makes the research more exploratory in nature and introduces several important limitations that should be considered when interpreting these findings.
Interesting.
So let's take a look at the methods. This was a non-experimental cross-sectional study involving 52 competitive male iron sports athletes. Participants were categorized based on their primary training of focus, either powerlifting, bodybuilding, or strongman, and further classified as anabolic androgenic steroid users or non-users based on a recent self-reported use. All the data was collected in a single session where the athletes completed detailed questionnaires covering questions relating to training volume, training style, and anabolic steroid use, including the compound type and dosage. Importantly, just as a quick side note, the bodybuilders reported significantly higher training volumes than the powerlifters and strongman, confirming that the sports classifications reflect meaningful differences in training approaches. Saliva samples were then collected and analyzed using quantitative PCR to assess mRNA expression of IGF-1, MyoD, and myogenin markers. Expression was quantified using cycle threshold values, where lower values indicate higher relative gene expression. And just as a reminder, these measurements reflect relative gene expression in saliva, not muscle tissue, and therefore should be interpreted accordingly. Now, despite these clear differences in training volume, the expression of IGF-1, MyoD, and myogenin measured in saliva did not show consistent separation or differences between the groups. Across all 3 markers, athletes who reported anabolic steroid use did not consistently display higher or lower gene expression compared to the non-users. Likewise, bodybuilders, despite higher reported training volumes, did not show clear differences in expression levels compared to powerlifters or strongman athletes. Now, there were some trends in the raw values, with some groups appearing to have lower cycle threshold values, meaning higher relative expression. But these patterns were not uniform across markers or training styles. In other words, no single group consistently stood out across IGF-1, MyoD, and myogenin. Taken together, these results suggest that even with meaningful differences in how much athletes trained, and despite differences in reported anabolic steroid use, these factors were not clearly reflected in the salivary gene expression measures captured in this study. So what does this all mean? Well, first of all, there are several reasons why these results should be interpreted carefully. And this is where folks on social media have gone a little rogue with their interpretation of this study. First of all, markers like MyoD and myogenin are known to fluctuate in response to recent training because the salivary sample collection wasn't standardized relative to the subject's last workout. Short-term acute changes could have influenced these measurements. Second, these were experienced athletes with varying training histories. Differences in how long individuals have been training, how recently they've competed, or how their training programs have evolved over time could very well introduce a substantial amount of biological variability. And third, while saliva offers a non-invasive way to explore molecular signals, the biological relevance of these particular markers in saliva, especially MyoD and myogenin, is still a highly emerging area of research. Simply detecting them in saliva doesn't necessarily mean they reflect muscle-level adaptations in direct or proportional ways. In simpler terms, just because something shows up in the saliva doesn't mean it accurately is reflecting what's happening at the muscle fiber level. These markers are much more meaningful when measured directly, and saliva-based measures are still very new and not yet very well validated. That said, the authors acknowledge several important limitations that are largely inherent to studying this population. They point out that this study was a snapshot at a single point in time, observational in nature, and this limits any inference about cause and effect. Training practices and steroid use were all self-reported. The training history duration wasn't controlled, and sample timing relative to the most recent exercise bout was also not standardized. And perhaps most importantly, they point out that the use of salivary mRNA introduces uncertainty about how closely these measures relate to the muscle tissue process. Now, this doesn't invalidate the approach, but it does mean the findings should be viewed as exploratory rather than definitive, a key distinction that's largely been overlooked in many online interpretations of this study. So, what's the key takeaway? Well, overall, this study tackles an important and technically challenging research question in a population that's rarely examined directly. Now, while these findings don't meaningfully change our understanding of hypertrophy, training style, or anabolic steroid use in trained athletes, and this is one of the reasons I almost didn't want to share it after digging into the methods, they do highlight just how difficult it is to study these topics in real-world settings. In that sense, this paper is best viewed as a thoughtful step towards addressing a complex problem, one that highlights the need for future research with larger sample sizes, longitudinal designs, basically where the same individuals are followed and measured over time rather than addressed in a single time point, and where possible used more direct measures of muscle adaptation. Well, everybody, thank you so much for watching. If you appreciate this cautious, balanced discussion of strength and hypertrophy research, especially in areas where the science is still evolving, please consider liking this video, subscribe to my channel, and share it with someone who enjoys evidence-based training discussions. Thanks so much for watching, and I'll see you in my next video.
Money is power, power is you. Stop messing with your mind just play.